Let's go. Now we were speaking about cell line. Let's go to media. What kind of criteria the cell culture media should fulfill to avoid scale-up problems? Free of hydrolysate and proteins. I go back again. The topic today is scalability, but I am speaking about hydrolysate, what that means. Normally, when we develop the process in small scale, we have a high soil lot, and we start working with this high soil lot. Process development takes 6 months, and in 6 months, we use the same high soil lot, more or less. Many cell culture media today, unfortunately, contain at least high soybean peptone. And then we transfer the process from small scale to large scale. In large scale, we have bigger lots. Within one campaign, it happens that the people change the lots. But we know that high soy is a risk potential in medium because lot changes influences the productivity. And then we discuss with management about scalability and unstable process. But this problem might come from the high soy. Therefore, the question is, why should we take out from medium? Why shouldn't we take out from medium? It is possible. We have demonstrated that. And the same also with proteins. And I don't need to repeat. And we have here a filter issue. Now, let's speak about water. Water to make the cell culture medium. Some companies use ready-to-use medium, liquid medium. How much time do we have?
Yes, 30 minutes left.
Very good. Some companies use ready-to-use liquid medium. However, many companies have already powders. They solve this powder in water and make the medium and develop the process. In small-scale lab, where we develop the process, some labs have these filter cartridges to make highly purified water and to use this water for media preparation. And then, when you go to 10,000 liter bioreactor, you have your own facility, own machines to generate this water. And the quality of water in your small-scale lab and in large-scale lab might be different, especially in terms of trace elements. Trace elements like aluminum or something, or nickel, whatever, which is really, really extremely low concentration happens in medium. And therefore, more or less, the scalability issue is then programmed. And in our lab, what we do is we go to production facility and pick up from there 500 liter medium every week. We solve our powder in the same water which is used later on in the production. In that way, we guarantee that later on, when the process is transferred, the production people use the same water. Filter. I think everyone uses the same kind of filter. If I use from Millipore or whatever from Zatur a certain filter type in small scale to filter my medium, of course, it is very logical to use the same filter material in large scale also. But what about with liquid to filter area ratio? In small scale, when we make medium, maybe we make 10 liter medium or 20 liter. And then we have a filter. And this filter is either over-dimensioned or less dimensioned. That means the filter might get some components from medium. We have spoken about insulin. But what about with some other charged ions, trace elements? And they can also retain these components. As soon as you go to large scale, you have a big filter, but you have also a lot of medium to filter it. Is the ratio between liquid and filter area still the same like in small scale? suppression of metabolic waste production. That is really a surprising statement. That relates to cell culture media development, and that relates to scalability. What is the logic here, actually? The logic is the following. Now, during my career, when I transfer the processes from small scale to large scale, in every process with my colleagues, some of them sitting here, we were discussing non-stop about pH regulation. The dead bands, how and when to add carbonate base into the culture, and how to regulate the pH. The pH regulation between small scale and large scale can be also different. The reaction time can be different, and so on. And because it is a problem, I thought, what can we do to solve this problem, to make it easier? The first question was, why do we add base into the culture? Because cells produce acid. When they produce acid, pH goes down, and we have to keep the pH in a certain area, so we add then base. Okay. The next question was, why these cells produce acid, actually? We don't know why, but they produce. That means by adding base into the culture medium or into the process, we are fighting the symptom, not the problem. The problem is acid production of cells. The problem is not unstable pH. And therefore, we went back and thought about acid production. What can we do to avoid acid production of cells? Acid production means the most prominent acid is lactate. We know in the last 30 years, I don't know how many hundreds of papers are published about lactate. Cells produce lactate, we know it. And we know also there are many publications. Lactate production is an symptom of overflow metabolism of cells. Because we have a lot of glucose, because of much glucose, cells produce lactate. If we should avoid lactate production, what should we do? Normally, we should limit glucose down, and then the cells would not produce lactate. That is published in many papers. Here, we have found a solution without glucose limitation to avoid lactate formation. And then after finding that, we have done these fundamental experiments. That is more or less the last experiment of many, many experiments. We have done following. We have taken a CHO cell line and put that in 3 different culture medium, 1, 2, 3, basal medium. And we have run the processes in batch. And we have not controlled the pH. We just monitored what happened in different media. What we see is in 2 media, the cells produce acid and the pH goes down. In another medium, the cells even consume acid and pH goes up. And after seeing that, the problem was solved. The solution was just balance the metabolites so that acid production and acid consumption remains in a balance during the whole process. If I can do that, then there is no net lactate production or no net lactate consumption. In this case, my pH should remain constant. That was the philosophy. And then we went to bioreactor. Here, 5-liter bioreactor of an antibody process. And that are the pH limits. And we ran the process that is offline pH measurement, and we could demonstrate it. It is really possible without any base addition to keep the pH within the ranges. That means for large scale, it is big trouble is gone, actually. We don't need base and all the discussions we can save. Now, that is small scale run. I was very interested to see that it also happens in large scale. 4 years ago, we have developed an antibody process. That is our first antibody process based on our platform technology. And we have transferred that from 5 liter directly to 2,500 liter bioreactor just to get these metabolic data. Yeah. And what we see is here viable cell concentration. And here, in terms of viability, I would say there is no significant difference. And in terms of product concentration, no significant difference. And that is now an important slide. It really doesn't matter in which scale the cells grow. And the lactate formation was similar. And the most important slide for me was this one. That is now the pH, offline pH. Online pH looks better. That is offline, 5 liter and 2,500 liter bioreactor. I was happy to see this confirmation. That means it works. And we don't need to discuss with our clients about pH regulation. Now, this slide could be understood so that it is one particular process. We were maybe lucky to get such a pH profile and lactate profile. Yeah, what about with the rest of the processes? I can confirm that in all our processes, we have this more or less same lactate formation and same pH progress, and we never control pH anymore. So